Welcome to the Circle of Life, an introduction to the cell cycle. Life, in its most fundamental form, is a continuous cycle of growth and division. This elegant choreography, played out within the microscopic world of cells, is orchestrated by the cell cycle. Like a spinning wheel, the cell cycle propels life forward, ensuring the growth, repair, and continuation of all living organisms. Through the precisely timed events of the cell cycle, a single cell gives rise to two identical daughter cells. This act of duplication underpins the development of a single fertilized egg into a complex organism and the healing of a wound. Each stage of the cycle is marked by specific checkpoints, ensuring that everything proceeds in harmony. Now, let's delve into setting the stage interface and its crucial roles. Before a cell can divide, it must meticulously prepare for the grand event. This preparatory phase, known as interphase, is often likened to the bustling backstage activity before a theatrical performance. Interphase can be further divided into three distinct phases: G1, S, and G2. Each phase plays a critical role in ensuring that the cell is ready to divide, equipped with all the necessary components for creating two identical copies of itself. During interphase, the cell grows in size, synthesizes new proteins and organelles, and duplicates its DNA. This intricate process of DNA replication ensures that each daughter cell receives a complete set of genetic instructions. Let's shine the spotlight on growth. Unveiling the G1 phase. The G1 phase, or GAP1 phase, marks the beginning of interphase and is a period of intense growth and metabolic activity. Imagine a newly formed cell, fresh from division. Like a factory ramping up production, the cell synthesizes new proteins, lipids, and carbohydrates. The cell also generates more cytoplasm, the jelly-like substance that fills the cell and houses its internal structures. During G1, the cell assesses its surroundings and internal state. If conditions are not favorable, the cell can pause the cycle, entering a resting state called G0. Section 4. DNA replication, the choreography of copying life. The S phase, or synthesis phase, is the star of interphase. It's during this phase that the cell undertakes the remarkable feat of duplicating its entire DNA. Enzymes unwind the double helix of DNA, separating the two strands like a zipper. Each strand then serves as a template for building a new complementary strand, following the base pairing rules: adenine with thymine and guanine with cytosine. The result is two identical DNA molecules, each composed of one original strand and one newly synthesized strand. To prevent errors, the cell has evolved sophisticated proofreading and repair mechanisms that operate during and after DNA replication. Section 5. Preparing for division G2, the final act before the big split. With DNA replication complete, the cell enters the G2 phase, the final act of interphase. During G2, the cell continues to grow and synthesize proteins necessary for cell division. It also replicates its organelles, ensuring that each daughter cell receives a complete set of these essential cellular components. The G2 phase is also a crucial checkpoint, the final dress rehearsal before the big show. The cell verifies that DNA replication has occurred correctly and that all necessary proteins and organelles are present. If any errors or deficiencies are detected, the cell cycle can be halted, allowing time for repairs or adjustments. Section 6. The grand finale: mitosis and the dance of chromosomes. Mitosis, the culmination of the cell cycle, is a visually stunning spectacle, a carefully choreographed dance of chromosomes that ensures the equal distribution of genetic material to two daughter cells. Mitosis unfolds in a series of distinct phases: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, and the mitotic spindle begins to form. Metaphase marks the alignment of chromosomes at the cell's equator, each attached to a spindle fiber. Anaphase follows with the separation of sister chromatids, each moving towards opposite poles of the cell. Telophase, the final act of mitosis, witnesses the arrival of chromosomes at the poles and the reformation of the nuclear envelope around each cell, resulting in two identical daughter cells.